Molds typically have asexual reproductive structures that we call asexual vegetative fungal spores. We use this fairly detailed description to distinguish vegetative fungal spores from bacterial endospores. Bacterial endospores will resist the effect of disinfectants and desiccation and a wide variety of extreme environmental changes and still remain viable. That's in comparison to the vegetative fungal spores that are usually very effectively killed or destroyed by disinfectants, desiccation, and other extreme changes in the environment. Every single vegetative fungal spore is simply a little tiny piece or copy of the entire genetic material of the fungi that, if it lands on the correct proper surface, will start to grow into a whole new structure, either a yeast or a mold. We have all kinds of specialized names for different kinds of vegetative fungal spores simply based on their morphology. We have microconidia, which are very tiny, macroconidia, which are large club-shaped, sporangiospores, arthrospores, even blastospores. Notice that the blastospore is also called a bud. Many species of mold have a cycle that would be a sexual reproductive cycle. There are plus and minus strains of certain types of molds that when they get close together, they will fuse, combine all of their genetic material to form a new cell that has twice the amount of genetic material than the previous two different plus and minus strains. That first cell that's the result of the fusion of two sources of genetic material is called a zygote. That zygote will undergo cell division, grow into a whole distinctly different structure, produce spores that can grow and result in that essentially plus and minus strain once again. Now, not every species of mold can produce these forms of sexual reproductive structures. And in fact, usually the sexual reproductive cycle looks so different from the original that it took scientists quite a few years to figure out which was which, and which two, for asexual and sexual reproduction, go together for the same species. When we're creating the scientific names of the different phyla of fungi, we're going to add the suffix mycetes or mycota to the end of the root word. The suffix mycetes or mycota should automatically tell us that this is some type of fungi that we're working with. Now, please be aware that some of the images that I've used in the presentations about the molds and the yeast and various types of fungi are often the ones that are clinically significant. As such, some of these images of clinical infections with molds and yeast can be quite disturbing or fairly dramatic. If they do bother you, please don't hesitate to move on to the next slide or simply just ignore the images. Our first phylum to discuss is the Zygomycetes phylum. Zygomycetes include a couple of different genera that are clinically significant and at least one example that is of historic significance. Rhizopus is the common old-fashioned black bread mold. It looks a little bit like salt and pepper. Notice in the photo, almost these very slender lollipops with the black lolly at the top and the long slender filament holding it up. Well, they really aren't lollipops. In fact, these large, very dark black spheres at the ends of these different hyphae are sporangia. A sporangium is almost like a balloon or a little cap, and inside that little cap are thousands of sporangiospores. When the sporangium gets large enough and mature enough, eventually that sporangium will rupture or bust open, releasing all of the sporangiospores inside. Now, when great-great-grandma made bread with no preservatives, rhizopus was the typical kind of old-fashioned bread mold that would grow on unpreserved, freshly made bread that simply was allowed to sit and get a little bit stale and moldy, if that ever did happen. We used to use it a lot in teaching in general biology labs because Zygomycetes rhizopus has a sexual reproductive portion of its life cycle that's fairly distinctive. Now here we have an example of two different genera of Zygomycetes that do cause a clinical condition called rhinocerebral mucormycosis. Now remember, a mycosis or mycoses, mycosis would be singular, mycosis plural, is a fungal infection. And then we're going to use a specific term or word that tells us where is the fungal infection. So rhinocerebral, nose, and head. And then mucormycosis is referring to the fact that a species of the genus Mucor is actually growing in the tissue of the patient. Or we even have examples of rhizopus species, not the common black bread mold, but other species that also can cause this type of mucormycosis. A generalized name for this condition, we could call it zygomycosis, or we could call it mucormycosis, more specifically referring to the genus Mucor. Zygomycosis could be either Mucor or rhizopus causing the infection. Notice the top illustration. You can clearly see that there is a tremendous loss of tissue for these patients who are infected. The middle image, you can see the long filaments of the mold growing in the tissues. This is actually a histologic section that's been stained. And all the matrix surrounding are the cells from the patient. But those long little filaments are not supposed to be there. That's the mold growing in the flesh or the tissue. And then the bottom photo shows you a comparison of an endoscopic image on the left and a radiograph on the right. Notice the endoscopic image on the left, the top photo, you can clearly see sort of the brownish, blackish tissue that essentially is infected with the mold, compared to the lower photo on the left of the normal, healthy turbinates or sinus passage. When we take a look at the radiograph, essentially the left-hand image, if you look carefully where the turbinates should be and the nasal passage, we actually see that there's some kind of mass. But we can definitely see there's some kind of inflammation around the turbinates because there's a much thicker area of tissue in comparison to the turbinates and the nasal passage on the right. Now, the first image of this kind of radiograph, the first thing you think of is that there's a mass in this location. And of course, when we think of some kind of mass growing or in the wrong place, the first thing we think of is a tumor. But in this illustration, in this example, it's not a tumor that's growing. It's instead, unfortunately, the mold growing and causing the problem. Now, if left untreated, of course, this is a very serious condition and is often difficult to treat. We often find patients who develop fungal infections already have pre-existing compromising conditions or immunocompromising conditions. Their immune systems just aren't working as best it possibly could because the patient is already sick with something else. So we see examples here of rhinocerebral mucormycosis in diabetic patients. And the diabetes has essentially caused all kinds of circulatory problems and other pre-existing problems, so these patients are far more susceptible to these fungal infections.